Hello everyone. Welcome to another video in the series of lectures on physiology of nervous system. For the earlier video, please refer to the link in the description box. We start with receptors. Receptors are sensory nerve endings that terminate in the periphery as bare, unmyelinated endings or in the form of specialized capsulated structures. Receptors give response to the stimulus. When receptors are stimulated, they produce a series of impulses which are transmitted through the afferent nerves. They are often defined as the biological transducers which convert various forms of energy into action potentials in the nerve fiber. On to the exterior receptors. The exterior receptors are further classified into three types. Cutaneous receptors that are present in the skin, chemoreceptors and telereceptors. Cutaneous receptors are otherwise called the mechanical receptors or the mechanoreceptors because they respond to mechanical stimuli such as touch, pressure, cold, temperature and pain. Chemoreceptors are those receptors which respond to chemical stimuli such as taste and smell, while the telereceptors are those receptors which react to stimulus from far outside the body. Hence they are called distant receptors. An example is vision, hearing. Interoreceptors are classified into visceroreceptors and proprioreceptors. Visceroreceptors are those situated in the viscera, while proprioreceptors are those which respond to change in positions of different parts of the body. Stretch receptors, baroreceptors, chemoreceptors, osmoreceptors are examples of visceroreceptor, while muscle spindle, Golgi tendon organ, Pacinian organ, free nerve ending, hair cells are examples of proprioreceptors. Synapses. Synapse is a junction between two neurons. These are not anatomical continuation, but just a physiological continuity between two nerve cells. Synapse is classified by two methods: the anatomical classification, based on whether the synapse is formed by an axon of one nerve ending onto the cell body, dendrite, or axon of the next neuron. Functional classification is based on the mode of impulse transmission. Anatomical classification of synapses is based on whether the ending of axon is on the cell body, dendrite, or the axon of the next neuron. When axon of one neuron terminates onto the axon of another neuron, it is called axo-axonic synapses. When the axon of one neuron terminates onto the dendrite of another, it's called axodendritic synapse. When axon of one neuron ends on the cell body of another neuron, it is called axosomatic synapse. Functions of synapses include transmission of impulses from one neuron to another and also inhibit some of these impulses. Based on the functions, synapses are divided into two types: excitatory synapses that transmit the impulses and cause excitatory function. Inhibitory synapses are those which inhibit the transmission of impulse and cause an inhibitory function. Based on the mode of impulse transmission, functional synapses is classified into electrical and chemical. The physiological continuity between the pre and post-synaptic neurons is provided by a gap junction, and there is a direct exchange of ions between these two neurons through the gap junction. As a result, the electrical impulse directly moves between the post and pre-synaptic neurons, and the action potential reaches quickly without any loss of time. There is minimal delay or no delay due to the flow of current. The impulse is transmitted in either direction through this electrical impulse, both ways. Cardiac muscle fibers, smooth muscle fibers of intestine, and epithelial cells of the lens in the eye are some of the examples where electrical impulse transmission originates. Chemical synapse is the junction between a nerve fiber and a muscle fiber, or between two nerve fibers, through which the signals are transmitted by release of chemical transmitters. There is no continuity between the two neurons because of the presence of a space called synaptic cleft. Action potential reaching the presynaptic terminal causes release of a neurotransmitter substance from the vesicles in this terminal. This neurotransmitter reaches the postsynaptic neurons to the synaptic cleft and then causes a change in action potential. Excitatory and inhibitory process. The release of an excitatory neurotransmitter at the synapses will cause an inflow of positively charged sodium ions, which results in depolarization of the membrane. Whereas inhibitory synapses causes an inflow of chlorine ions and outflow of potassium ions, resulting in hyperpolarization and this in turn will prevent the discharge of impulses. Summation is defined as the fusion of effects or progressive increase in the excitatory postsynaptic potential in the postsynaptic neuron when many presynaptic excitatory terminals are stimulated simultaneously or when single presynaptic terminal is repeatedly stimulated. Summation is of two types. Either spatial summation occurs when many presynaptic terminals are stimulated simultaneously. Temporal summation, which occurs when one presynaptic terminal is stimulated repeatedly. Both the types of summation can happen simultaneously as well. Neurotransmitters. Neurotransmitter is a chemical substance that acts as a mediator for transmission of nerve impulse from one neuron to another neuron through a synapse. For a substance to be called as neurotransmitter, it has to be found in the neuron, it must be produced by a neuron, it must be released by a neuron. After release, it must act on a target area or produce some biological effect. After action, it must be inactivated. Neurotransmitters are classified depending upon their chemical nature or depending upon their function. Many chemical substances can be identified as neurotransmitters. Examples of amino acids. Amino acids are fast synaptic transmission neurotransmitters. They can be inhibitory as well as excitatory in action. Glycine, GABA, glutamic acid, and aspartic acid belong to these group. Amines are modified amino acids. These involve in slow synaptic transmission. They can be inhibitory as well as excitatory. Noradrenaline, adrenaline, dopamine, serotonin, and histamine belong to this group. Some neurotransmitters do not fit into either of this category. For example, acetylcholine, that is categorized as others. The excitatory neurotransmitters are those which cause excitatory action. They end up causing what is called as an excitatory postsynaptic potential, EPSP. Inhibitory neurotransmitters are those responsible for inhibitory action. They produce energy called inhibitory postsynaptic potential. Thank you for watching this video. Wait for our next video to continue further on this topic. If you found this video useful, please do share with your friends. If you had feedback, please leave it in the comment section. Happy learning.